joining this. So my name is Alex Doney. I'm a senior lecturer and consultant physician at Ninewells Hospital and Medical School. And this is what I'm going to be telling you about today: investigator-driven clinical trials for development of precision medicine approaches. So this is largely my experience and my view of the way we are doing these sorts of these issues in my university. And I think they apply and will apply really to the future of precision medicine, and very much how we are going to do this in a global context. So I think maybe I should, is my picture showing in the middle of the screen there? Maybe I should just get rid of that.
We're seeing the whole slide. We're seeing your slide actually, like your presentation.
Okay, but you're not seeing my face in the middle of it, which I'm trying to hide because of, good. Okay.
So you can maybe like catch it with the arrow and move it if it's messing with your.
Okay, I've done that.
But we don't see it. Yeah.
You don't see it. That's good. Okay, so I mean, this is the essence of precision medicine and the way we think about it. A lot of precision medicine is based on genotyping, and we might consider genotyping the germline, in other words, the DNA that we transmit through generations. But also a lot of precision medicine is based around cancer therapy and looking at somatic mutations within the tumor itself. And so there's a large amount of information from the genome from within the somatic DNA, but also increasingly we're interested in the microbiome, in other words, the vast number of microorganisms that inhabit either on us or in us and how they affect our health and disease. And so this is becoming another very important area of genotyping. But one of the most important aspects of doing clinical trials in precision medicine is precision phenotyping. And with increasing technology in terms of imaging, plasma biomarkers, tumor biomarkers, but also electronic medical records, we're gradually able to assemble more and more detailed phenotypes about the clinical diseases that we want to investigate and manage. And that's a critical, important part of developing trials. So that's the essence of really what precision medicine is. And I'm sure many of you will have seen slides like this where we have a diseased patient population. I hope you can see my cursor moving on the screen. And yes, we see it. And then we have this current standard approach to treatment, which is the way people who practice medicine, such as me and others, have been doing for many years, is that we don't know about all the different types of individuals that we can phenotype or genotype within this population. And we give them a single treatment. And that treatment is only effective in about 20% of the population, with many individuals not responding to that treatment. But what we hope to do with precision medicine is to divide the diseased population up into these groups of individuals, and they will respond either to one treatment or another particular treatment. So on the one hand, just as I was saying previously, we're using phenotyping to define individuals, and then we are treating individuals with different treatments. And this lends itself to the concept of theranostics, which is a word coined by the Americans, but I think it's a very valuable word because it tells us that in the same instant that we are making a precision diagnosis, we are deciding on the precision treatment that individual will receive. So that's the concept, but there are some issues around that and some things we need to be aware of, because medicine is much more, precision medicine is much more than just diseases and treatments and getting evidence for that. It's a very, very wide process of healthcare. So yes, we can use genomics, genetics, physiology, digital phenotyping, etc. But all of this feeds into platforms for being able to change clinical care guidelines and pathways, but also to activate decision support so that we can provide healthcare and also information to different members of healthcare providers, the clinicians, but also patients, but perhaps also caregivers as well. So it's a much more complicated process in terms of delivering efficient healthcare, and trials, clinical trials will need to be cognizant of that. So as everyone knows, evidence-based medicine is based largely on the gold standard of the randomized placebo-controlled trial. And this is the science that we undertake doing these randomized placebo-controlled trials. And this leads into guidelines, and the guidelines affect doctors' behavior and subsequently improve patient care. But the trouble with this model of delivering evidence and trial evidence for healthcare is the randomized placebo-controlled trial is expensive. It's a very expensive thing to do. It's a very time-consuming thing to do. It's very highly regulated with issues around consent and obtaining ethics. It has very poor external validity because the sorts of individuals that get enrolled into clinical trials are not really representative at all of the kind of patients that we see in everyday clinical practice. And so the evidence that we generate from clinical trials doesn't necessarily apply to the patient population that we want to treat. And essentially, what we have when we are thinking about the randomized placebo-controlled trial is we're looking at the average effect of a drug in an average population. So it's not really conducive to individuals within that population. And there's certainly not enough, even for the medicine that we currently practice. We don't have enough clinical trials to be able to provide us with all the evidence we need. So that's one of the big problems that we have in medicine already based on the clinical trials that we do. And the problem with a precision medicine approach to clinical trials is these problems are magnified many fold. So if you take a typical patient population, like patients who have problems with raised blood pressure, for example, that's just one disease, this ray of light coming in here, just one disease. And that's the disease that we do trials for and we manage conventionally. It's a single entity that we think we understand clinically. But as we do precision medicine, we're going to get a spectrum from this particular single disease, where, as I said earlier, you have different treatments for different subsections of that population. And this creates problems because it fragments the disease into many different populations. All of these different treatments are going to require regulation and evidence for their implementation in healthcare. They're all going to have to be subject to evidence generation, in other words, clinical trials around those. And then doctors, in terms of the medical model that I showed earlier on, in terms of guidelines, etc., are all going to need to be educated and provided information for each one of these things. And as I said before, it's already very hard to do that with the evidence that we're generating. And so precision medicine is going to simply amplify the problems that we've had. So that we do need a way of answering questions more efficiently and in less time. And that's really what I'm going to be approaching in this talk to you today, in terms of different different approaches and different thoughts for being able to do that. So cancer medicine has been the main driver for doing precision medicine studies, principally because cancer is, a lot of the medications that we use for cancer are very dangerous and can cause harm. And so the therapeutic range for these drugs is very low. The risk-benefit ratio is a critical issue. So because the cancer medicines can do so much harm, but also there's a lot, we understand a huge amount about the biology of cancers themselves in terms of genetic variation of the somatic mutations that cause the cancer to evolve and develop. And so many of the paradigms for precision medicine come from cancer medicine. And these are the main types of clinical trials that have been developed for being able to cope with the sheer number of potential studies that need to be done and the amount of evidence that needs to be generated. And I'm going to tell you a little bit about these just now. But essentially, this is a collection of trials that share key design components to achieve better coordination across them so that you can get much more evidence much more quickly in a shorter period of time in a much more efficient manner. So taking adaptive trials, just first of all, I'll tell you a little bit more about these. So in an adaptive trial, there's a continual modification as you go through the trial based on the trial design, allowing you to look at the process of the trial as it's happening, gather more information as you're progressing through the trial, and modify the trial as you go forward. And this approach reduces the use of resources and time. It's much more efficient, and it also improves the likelihood of success. So for example, you can reassess the sample size. So most conventional trials, you have a fixed sample size and you undertake the trial based on that sample size. But one of the things that adaptive trials aim to do is do an interim analysis and then change the sample size calculation based on data that you gain from the earlier parts of the trial to really make sure that you have sufficient power to finish the trial. So that's one aspect of adaptation as you go along. You can also adapt the allocation ratio, the number of people that you put in the cases or in the control group, based on the evidence that you generate as you move through the trial. And this ensures that more patients can receive a more superior treatment as you go forward. In other words, it doesn't jeopardize patients who may benefit from a treatment as the trial matures and more evidence becomes available, indicating that certain people are actually benefiting. You can drop inferior treatments in treatments that don't seem to be working very well, and you can add new treatments in as you move forward. And this can also save time and resources. So this is again adapting as you go through the trial. And you can also enrich the population within your trial to make sure that you've got the right balance of risky people that are probably going to benefit from the treatment. And one of the other important things is through the different phases, phase 2 and phase 3 of a clinical trial, you can actually transition within one single trial framework from a phase 2 clinical trial to a phase 3 clinical trial, again, making things much more efficient and much more effective, tying things together, making things efficient. But of course, with all of this does require extensive planning and simulations of the trial data, of the likely trial data that you're going to gather, so that you can be absolutely sure of the consequences and gains of all the possible adaptations that you will make and change the decision rules. And again, the statistical analysis plan for those trials needs to be fairly convincingly drawn up in advance. So here's just a cartoon essentially of what I've just been talking about, the adaptive clinical trial. So this is, at the top here, is a conventional clinical trial where you have a treatment and you have standard of care, which is the placebo arm. You perform an interim analysis to look at the data, but you still stick with your original planned sample size with these number of individuals in the control and the case group, or the treatment and non-treatment group. But with an adaptive clinical trial, you'll have a sample size reassessment halfway through the trial or at some stage during the trial. And as a result of that sample size reassessment, based on the evidence gained through this part of the trial, you can add more patients in to increase the sample size, to increase the likelihood of detecting the signal that is the basis of your clinical trial. You can also, for example, in an adaptive trial, you can change the number of people that you allocate to a particular subgroup. So in this particular example here, more people have been added to the intervention group, the treatment group, in order to allow these individuals to benefit. And again, with an adaptive trial with a sample size reassessment, you can put more of different types of people into your clinical trial to allow comparisons to be made of the many different types. So there's an example of being much more flexible, but it does require quite a lot of planning and statistical analysis, both during and after the clinical trial. And two other approaches that I mentioned earlier on are the umbrella trial and the basket trial. With an umbrella trial, it's a single disease, for example, like hypertension or a single cancer. And here you have many targets that you're looking for in order to do precision medicine. And you can undertake each one of these as a separate sub-trial, if you like. So biomarker 1 and targeted therapy 1, biomarker 2, targeted therapy 2, et cetera. So in other words, you have many smaller trials underneath this one large umbrella trial relating to a single disease. The converse of that is the basket trial, where you have many interventions, but all targeted on many diseases, I should say, but targeted on one intervention, which might be relevant to each one of these diseases. And both of these trials, I suppose, can be part of a whole platform of trials going forward. These platform trials, which here's an example of a single disease, whereby you're adapting the whole time that you're going forward through time in a whole program of carefully designed work. For example, here for this single disease, you have these different biomarker substrata, and you decide on different drugs that you're going to look at. But as you go through time, with more information, a new drug might be added, or you might remove a drug or stop a particular arm of it due to futility, for example. So here you have a whole program of work all knitted together in one comprehensive, overarching study design. And this gets away from this slow, one-by-one trial approach, which is inefficient and ineffective. One of the most important aspects, clearly, about thinking about trials is the statistical concepts that you bring to bear in relation to deciding how you're going to analyze and conceive of your clinical trial. And that depends on the main research question and what is the appropriate analysis for the particular hypothesis that you have. So for the standard randomized placebo-controlled trial, the emphasis in those trials is largely on trying to demonstrate superiority. In other words, compared to the placebo, the study drug, or the drug that you're interested in, or the intervention that you're interested in, you want to prove is superior to the placebo arm. And that tends to be a much more conservative approach, and an intention to treat approach is taken, because really the attempt here is to avoid a type 1 error. So you really want to prove that your intervention is superior to the non-intervention arm. And that's due to things like non-compliance and missing outcomes. However, in many of the trials that we've just been looking at, we're much more interested, not so much on superiority, but whether they're equivalent, but for which you get other benefits. So we want to show that treatment A is equivalent to treatment B. In other words, both treatments are active treatments, but by using treatment B, you're able to get some other benefits, like, for instance, greater safety, it's more convenient, it's more cost effective, it's more available, et cetera, et cetera. So in many of the precision medicine trials, rather than necessarily being concerned about superiority, we're more often interested in being equivalent or non-inferior. And these tend to use anti-conservative approaches, such as per protocol and as treated analysis. So just on that subject, when we're thinking about comparing therapies and looking at for non-inferiority or equivalence, there are some major differences philosophically in how we consider these trials. So as I said, in a traditional comparative trial against placebo, the null hypothesis is that there is no difference between the therapies, and the research hypothesis is that there is a difference between therapies. However, for equivalence and non-inferiority trials, it's the other way around. And the research hypothesis is that the new therapy is equivalent to the current therapy, and the null hypothesis is that the therapies are not equivalent. And it's this equivalent margin, or the margin of equivalence, that I'm showing over here, whereby you want to be able to show that they are equivalent within a boundary of this non-equivalence, or this equivalence margin, or non-inferiority margin, which is a critical aspect of demonstrating that that your therapies or your therapeutic approaches are equivalent. So these are things that emerge from precision medicine trials, as opposed to typical trials of clinical effectiveness. So to be able to illustrate some of these concepts, I'm just going to quickly talk about a particular precision medicine trial that was published last year. And in order to make sense of that, I'm going to just quickly cover some ground around the precision medicine approach there. So for those that don't know, and I'm sure many listening do know, clopidogrel is a drug that's used very widely across the world to prevent clotting, or to prevent thrombosis. And it's widely used by cardiologists and stroke doctors to stop arteries getting blocked up with clots. The thing about clopidogrel is it's a prodrug, and that means that clopidogrel needs to be metabolized in the liver once you swallow it, in order to be activated into its active components. And about 25 to 30% of people in the world, so 25% of all of you people listening to this talk, carry a genotype that means that they are less able to metabolize clopidogrel, and that means that giving them clopidogrel, the clopidogrel doesn't work as well in them as it does in the rest of the population, the 75% of the population who metabolize clopidogrel well. And it was because of this that in about 2010, the Food and Drug Administration in America put a boxed warning on clopidogrel, indicating that in patients who are poor metabolizers of clopidogrel, an alternative should be sought. So that's a little bit about the background. And so you can imagine a precision medicine study where you want to determine if having knowledge about whether you're a clopidogrel slow metabolizer or not there might be a treatment that you can get that doesn't put you in harm's way simply because of your genotype. And this is the problem, because in a nutshell here, if you tend to clot, you cause the blood to block a blood vessel, and that can cause a heart attack or a stroke or something equally horrible. Whereas if you give people an antiplatelet drug like clopidogrel, it stops this clot from forming, but it can also cause bleeding, because if you're not able to form a clot, when a blood vessel is damaged, as in this cartoon here, when a blood vessel is damaged, if you can't form a clot, then blood escapes and you bleed. And so when we're thinking about treating people with clopidogrel, or indeed any of these other drugs, the problem is that you can either cause bleeding, or if you don't use enough of these drugs, or these drugs aren't working, you can cause, or the clot will reform, and so you're in this constant balance. So this particular study here that was just published in the New England Journal last year is an interesting one, because what this study was asking was whether a genotype guided strategy for these particular drugs, these P2Y12 inhibitors, of which clopidogrel is one, might improve the management of patients who are slow metabolizers of clopidogrel. So this is the overview, and it's just important because it'll lead on to one or two other things that I want to tell you about in the next few slides. So this was the name of the study, Popular, and it was a randomized open label assessment blinded by the assessors in patients undergoing primary percutaneous angioplasty with stent implantation. So these are stents to keep the blood vessels in the heart opened up. And they were randomized 1-to-1 to receive either any one of these drugs on the basis of their genetic testing, or standard treatment with these standard drugs. So in the genotype guided group, these were people who had loss of function of these cytochromes, which are in the liver, which metabolize. So in the slow metabolizers, in other words, people for whom clopidogrel wouldn't work, they received alternative drugs, ticagrelor or prasugrel, whereas non-carriers received clopidogrel. In other words, if they were normal metabolizers, they received clopidogrel. And this is how the study was conducted. 2,751 patients underwent randomization, and half of each, about 1-to-1, were assigned to the genotype guided group and the other to the standard group. This was how the trial was followed through, up to 12 months of follow-up. And here are the primary outcomes that the study was interested in. The first primary outcome was net adverse clinical events. In other words, death from any cause, a myocardial infarction, definite stent thrombosis, or stroke, or major bleeding. So in other words, a composite endpoint of all of those things, a net, all the various clinical events. So in other words, including infarction, in other words, thrombosis or bleeding. Whereas the second primary outcome was major or minor bleeding at 12 months. And their primary combined outcome was tested for non-inferiority. That's an important part of this particular study and how it differs from conventional randomized control trials. And here they used a non-inferiority margin of 2 percentage points for the absolute difference in the two study arms. And they did two approaches to the analysis. One was an intention to treat, but they also used an anti-conservative approach of a per protocol analysis. So in terms of the outcome, the primary combined outcome, there was an absolute difference of minus 0.7 percentage points, which was significant for that non-inferiority margin. So in other words, using genotype guided approach was non-inferior to the conventional approach, but that was bought at the benefit of reduced bleeding, a significant reduced bleeding in the approach that used the genotype guided approach. So here you have an approach or a study that's shown that there is equivalence. In other words, you have one treatment isn't of any benefit compared to the other, but you have reduced chance of harm being caused as a consequence of doing a precision medicine approach. The other important thing to say also here is that clopidogrel is much cheaper than these other drugs, which are still under patent. And so if you can, that means that for 75% of patients, you can give them the much cheaper drug, and it's going to be equivalent to the more expensive drugs. So that's a good example of why in precision medicine, you might be more interested in an approach that's looking at equivalence rather than superiority. Yes, so that was the conclusion as I've just stated it. The genotype guided approach was non-inferior and resulted in a lower incidence of bleeding. So one of the things that I think is important when considering doing clinical trials in precision medicine is that larger and larger numbers of people now are taking part in biobanking and resources for being able to do precision medicine research. So good examples of these are, I'm sure you will have all heard of the UK Biobank, which is 500,000 people who have genotyping. Increasing number of these are being sequenced now, but also clinical phenotyping linked to electronic health records, but also clinical imaging, etc. And Genomics England is another example of increasing numbers of individuals in healthcare who are being genotyped in order to be able to deliver precision medicine approaches within the healthcare setting. And in Scotland, we have a program called SHARE, which is individuals providing consent to collect their blood and to link their medical records to their genetic information to be able to do healthcare research. And one of the very interesting approaches that's gradually coalescing out of all of this enterprise is that the research aspect, in other words, deriving information about precision medicine paradigms that might be useful for improving healthcare, is taking place increasingly within the healthcare environment, such as SHARE and Genomics England. And so we have a situation here where we can generate evidence for clinical benefit within these bioresources, and also deliver healthcare improvements within these bioresources as well. In other words, research and healthcare are coming together. And one of the other very important aspects of being able to do this is because as the population that are being genotyped gets bigger and bigger and bigger, and more of the population are being covered by genotyping, greater amount of research and trials can be done. And this is in particular because the cost of genotyping and phenotyping is rapidly decreasing. So here is the cost of sequencing the human genome over the past 20 years or so, and you can see that it's coming down and down and down, and will continue to come down still further with new technological developments, which means that rather than a single genotype, the example of which I was showing you, for instance, in the clopidogrel genotyping for those two particular cytochromes P450 that were being genotyped, you have all of an individual's genetic information contained within their genome. And so you can do just like those trials that I was talking about earlier on, the basket and the umbrella trials and the platform trials. You can do multiple trials on multiple individuals at the same time, because they all have a whole wide range of different variants right across the genome that have been genotyped. But also we're increasingly able to precision phenotype based on large-scale computation, machine learning, deep learning, data linkage across increasing numbers of data resources in order to be able to do big data approaches to precision medicine. So in that context, I just wanted to point this particular study out. This is one, in fact, that we did in our group and just published a little while ago. And this was within a bioresource, but it embodies this idea of Mendelian randomization. So rather than randomizing patients to one treatment or another, individuals are by default naturally randomized to the genetic information that they've inherited. So individuals, in this case, individuals who are taking clopidogrel and were being admitted to an acute stroke unit in our particular part of Scotland. We as doctors didn't know what their genetic status was, and we just treated them with clopidogrel. But we were able to show that individuals who were slow metabolizers had a very, very high increased rate of stroke as a consequence of being slow metabolizers, as a consequence of simply being randomized at birth to being slow metabolizers. So in other words, you can do virtual clinical trials based on randomization within a study population. But then the next question is, well, how do you then demonstrate that you can just like in the popular study that we were looking at, how can you demonstrate that by intervening on those individuals, you can prevent future events? So if you think about it, this is the population out there in the real world, and what we've been doing within bioresources is capturing all of their information into this bioresource repository, where we analyze the data and we produce outcomes and papers and evidence based on the data that we're collecting. But one of the things that we don't do and haven't done well is being able to use that data to actually improve healthcare. And this is one of the main processes that we're trying to do in precision medicine, and the trials that we do, is that in the end, every individual, every healthcare interaction will be enhanced by access to large-scale data and advanced analytics. So here we have these individuals, and here we have this large-scale data and analytics. And the one thing that's missing is being able to use this data actually to care for the patients and people that have contributed to all of this data in a precision medicine approach. So in other words, if you have all the data over here, you should be able to use that data to undertake precision medicine. And that's a part of this predictive, preventive, personalized, and participatory approach that Leroy Hood commented on earlier on. And this whole cycle then becomes a learning healthcare system whereby individuals who are being, whose data is being used to develop knowledge are also benefiting directly from that knowledge constantly in a learning healthcare process, and you're generating new knowledge in terms of interventions down this path here. And so this is where clinical trials can be directed. So in one of the examples of that that we're doing in our group is individuals who have taken part in SHARE and given their DNA and their electronic medical records. We are extracting from here their clopidogrel metabolism status and providing that information to doctors. And so doctors can then intervene and provide those individuals with the right kind of therapy at the right time to stop a stroke from happening. And as a consequence of doing this, this randomized process, if you like, we're able to follow these patients up using exactly the same approach to be able to do a clinical trial whereby we can determine the outcome of this in exactly the same way that we determine the outcome in the randomized Mendelian randomization context. So I just wanted to say a little bit more about the Learning Healthcare Service. Essentially, what I've been talking about is exploiting routine healthcare, both activity and data, to answer important clinical questions. So in other words, doing the science and that process that I spoke about right at the beginning of this talk, which leads to evidence and then subsequently to patient care. There's quite a lot of failures along that path where the evidence is either poorly used or the insights are poorly managed or the experience is poorly captured and doesn't actually benefit patients. So there's a lot of missed opportunity and there's a lot of harm and waste that goes on. Whereas with the process that I've been talking about is this constant feedback from this back into this loop so that you have this positive cycle of research that's happening and producing more evidence, but then feeding back and cycling back to generate evidence and improved health outcomes. So one of the examples that we're working on at the moment is what could really be described as randomized epidemiology. And in this particular study, we call it the evidence study, we're randomizing general practices in the community to a prescribing policy. Now, in this particular study, where the policy is to prescribe one of these drugs, one or other of these drugs, but it could equally be a policy of interacting with someone's genotype. So in other words, one group of practices would prescribe clopidogrel to people who were normal metabolizers, and in another group of practices, they would prescribe ticagrelor, say, to people who had had a stroke and were slow metabolizers. And this randomizing general practice in this way to a policy very, very rapidly creates a very, very large study. So for example, 50,000 individuals can be recruited from just simply recruiting 250 practices. And furthermore, these individuals are very representative of the kinds of people that we are treating in healthcare anyway. So this cluster randomized study of prescribing policy, the example of which is these two particular blood pressure lowering drugs, could be exactly the same approach could be applied to a randomized policy of precision medicine, and would be a very rapid, very quick way of getting very, very large study numbers. And in the group that I work in, we have a reputation for doing these many, many similar studies of very, very large scale pragmatic, efficient trials exploiting everyday healthcare activity, which means that evidence can be gathered very, very quickly and rapidly, and is very, very representative of the kinds of populations that we treat in healthcare every day. So just to conclude with, I've taken you through many aspects of clinical trials and how these relate to precision medicine. But I think that the future, especially in terms of the number and scale at which precision medicine trials are going to need to be done, does mean we're going to have to think of very practical, pragmatic ways of capturing large swathes of the population very, very quickly to generate evidence rapidly, taking a leaf out of the cancer trials approach, which is platform trials, basket trials, and umbrella trials, but do these in a very, very pragmatic way, using electronic medical records and already existing data such as genomics from people who have taken part in large bioresources. And rather than very expensive trials where individuals need to come in to hospital, they can be done from home and remotely using really next generation of IT and communications, web-based, mobile phone-based, etc., to be able to do these trials very rapidly at home. So that's all I have time to say, I think. Thank you for listening. And I think there's going to be questions. I'm not sure.
Thank you. It was really a view in the future with the precision phenotyping and this adaptive clinical trials. Maybe you can comment what you think about using this adaptive trials for COVID-19 and like new diseases that could come up in the future.
Yes, so COVID-19 is a very good example, and we can see from all the information that we are bombarded with every day and in the literature that COVID-19 is a very good example of individuals who respond differently to both the disease itself and also to the response to therapy, so very much so the approach of being able to develop a whole adaptive trials platform for being able to rapidly develop a whole series of treatments in a focused, international focused way, which means that trials are run in parallel in a unified way, would be very definitely the future of being able to develop strategies, precision medicine strategies for reducing the impact of COVID. It would be a very, very good example. But usually these platform trials require substantial collaboration across multiple groups to be able to do that. And I think probably we are still in the early days of COVID to be able to elaborate such structures.
Yeah, I agree. Collaboration is actually very time consuming. And another question about this new approach that we use data to care for patients who actually contributed to research. Do you know like many examples where this is used already in your university, or it's like a very new approach?
No, it's a fairly new approach based on the fact that still the regulation around bioresources. So I suppose the point that I was trying to make is that as the cost of genotyping, for example, becomes lower and lower and lower, increasing numbers of people, you will be able to obtain genetic information very, very quickly and easily. But at the moment, there is a division between data that's collected for research and data that's collected in the clinical environment. And I suppose that the point that I was trying to make there is that gradually, certainly in our university where we are pioneering a lot of these thoughts, is that it's the same data. And I think that was the point I was trying to get across, is that it's, we are finding now that we can start taking data from these bioresources and actually using it in the healthcare context rather than simply locking it up inside a database. So that, for instance, as we genotype more and more people, if they have that data sitting in a data resource for research, why not also use that data in the context that a patient comes into hospital, and that piece of data would be useful for them. So these are concepts, I think, for the future. They're not happening on a wide scale now, but I think that in terms of planning for precision medicine within countries, for strategies going forward, combining the concept of biobanking with for research, but also direct healthcare delivery and trials is a very important strategy going forward.
Okay, thank you. Because it's a similar idea that we heard today from Estonian Biobank. They are really trying to implement this. And do you think there is a huge change also necessary in like medical education to move forward with this?
It's one of the biggest problems, and I suppose one of the reasons why I wanted to talk about the P4Me study that I'm doing here is that yes, the current generation of healthcare providers or deliverers, doctors, nurses, healthcare professionals, are very, very, I don't think the word is uneducated, but aren't aware of how this is going to impact their medical profession and their career in the future. And so we do absolutely need to educate a new suite of doctors, nurses, etc., to be able to understand the concepts of precision medicine. And to that end, we do have an MSc course that we are delivering now at our university, which is delivery of precision medicine in a practical way. Because without that education, and I suppose one of my slides right at the beginning was showing that healthcare and delivery of precision medicine is much more than about the simple concept of treat this group with this drug or that group with that drug. It's how you integrate that into the healthcare setting so that individuals are aware of it. Already as doctors, we don't make enough use of evidence that we generate, even for the studies, the number of randomized controlled trials that we do. And so how we're going to be able to do that for say, a chronic disease like blood pressure or diabetes with precision medicine, when we suddenly, instead of having one disease, we have 10 or 20 subcategories of disease. So we're going to need not just education, but also decision support, because I don't think any one individual is going to be able to understand all of that. And again, that's where big data approaches and decision support and computation will help doctors and nurses to be able to deliver the results of precision medicine trials in real time at the bedside or in the clinic. They're not going to be able to do it without also education, but also, as I say, decision support based on computational algorithms.
Yeah, I agree that from students of medicine and biomedicine that I know, that we are younger generation and we have this very optimistic view what is possible and like how everyone should operate and everything, but like the institutional change is also necessary, I think, and some people are stuck with
So one of the things I'm very interested in is precisely that that end of the precision medicine delivery research. In other words, yes, it's fine to have precision medicine trials which demonstrate equivalence or effectiveness of one precision medicine paradigm against another, but it's quite another thing to be able to incorporate that into the healthcare environment. And that's where I think, again, why I was somewhat laboring the point about learning healthcare systems, so that evidence generation is part and parcel of, is contained within the healthcare system, so that it's not separate from it, because then that does engender learning and understanding. And so you're being randomized to a treatment by the decision support algorithm that you're using at your desk. So you're learning, so you're also learning in an adaptive way to precision medicine as it's being introduced. So I think that aspect of precision medicine delivery in trials is fundamental. In other words, making it an intrinsic part of everyday healthcare activity.
Yeah, thank you very much. I am coordinator of this project, and at the third year of the project, we have a lot of discussions with the local, medical community and other stakeholders. So many ideas from your presentation to include in that. Thank you very much.